Hi there. I want to take a few minutes to talk about an important concept in my microbiology class, which is the impact of penicillin and another enzyme called lysozyme on peptidoglycan cell walls. So in a microbiology course, as we're learning about bacterial cells for the first time, we discuss a lot about the bacterial cell envelope. And so we're aware of these structures, that all bacterial cells have that cell membrane on the inside. And then just outside of that, they have this peptidoglycan cell wall structure. And then in some bacterial cells, like gram-negative bacterial cells, there is an outer membrane outside of that peptidoglycan cell wall. As we're learning about these structures, right, we want to think about the importance of the structure and how we could potentially use that structure as a target if we would like to harm a bacterial cell. So we know where the peptidoglycan cell wall is in terms of this bacterial cell envelope. And in addition, we know what the bacterial cell wall is made out of. It's made out of this chemical we call peptidoglycan. And so here's a simplified image of the chemical structure of peptidoglycan. There are these NAG monomers, and then there are these NAM monomers. And we get these long chains of alternating NAGs and NAMs. And we notice that the NAMs have this little chain extension off of them. These are some amino acids. And we notice that a NAM in one of these chains of NAGs and NAMs, that NAM can be connected to, cross-linked to, a NAM in another chain. And that cross-link is between amino acids that are part of the NAM monomer. So we know that the bond that connects amino acids together is a peptide bond. So there's this peptide bond cross-link, holding the NAM of one chain to the NAM of another chain. And that's how we can get this large interconnected peptidoglycan structure. So we know where the cell wall is in the bacterial cell. We know what the cell wall is made of. And so the last thing we should emphasize before we think about penicillin and lysozyme is what the cell wall function is. And the cell wall serves a few functions, but primarily, to me, it serves this critical function for bacterial cells. The bacterial peptidoglycan cell wall prevents death of the bacterial cell by a process we would call osmotic lysis, which is just a fancy way of saying popping or bursting. And so this diagram demonstrates a bacterial cell here with an intact cell wall. But if the bacterial cell has a defective cell wall for some reason, water will constantly be trying to enter this cell by osmosis. That will be happening with or without the functional cell wall. But if the cell wall is defective, the bacterial cell can no longer protect itself from the effect of this water entering by osmosis. So as more water goes into this cell, the cell gets larger. And here the diagram is showing that the cell is beginning to bulge out of where that cell wall was. The cell wall is supposed to be a strong container that keeps the cell from getting any larger than that cell wall structure. But when the cell wall is defective, the cell can grow in volume because of the entry of water and become larger than it should be. Now, if the cell gets too large, it will actually rip open, burst, and die. We call that process lysis. So the primary reason that bacterial cells have a peptidoglycan cell wall is to prevent the cell from dying from this lysis process. So we know what the structure is, we know where it is, we know what it's made of, and we know why the cell has it, to keep the cell from dying. There are times in medicine especially that we want to kill bacterial cells, perhaps because they're making a person sick. And so if having a weaker cell wall is a way that could result in bacterial cell death, perhaps we should investigate how could we target that structure to result in the death of cells. So, let's now jump into penicillin. Penicillin is a bacterial enzyme inhibitor. Penicillin inhibits this enzyme that we call transpeptidase. So the bacterial enzyme transpeptidase, and it goes by several names. Another name you might see for transpeptidase is penicillin binding protein, or PBP for short. That bacterial enzyme is responsible for creating the peptide bond crosslinks in that peptidoglycan cell wall structure. So let's walk through what penicillin does to this enzyme and how that results in a weaker cell wall that may cause the bacterial cell to die. So again, here we're looking at a simplified diagram of the peptidoglycan cell wall structure. As you can see on the left here, we've got different colored blocks to represent the NAG and the NAM. Sorry, I advanced one slide. So we've got a NAG, then a NAM, a NAG, then a NAM. We've got one chain at the top and one chain at the bottom. Again, there are these other symbols that represent these different amino acids. So off of the NAMs, we've got this amino acid chain. Off of this NAM, we have another amino acid chain. And then the last thing we're showing here currently is a representation of this transpeptidase enzyme. Again, it's also called penicillin binding protein. So let's watch what penicillin binding protein, or transpeptidase, is supposed to do. So this enzyme should approach this not yet complete peptidoglycan structure, and it should bind the amino acid chain connected to one NAM and create a new peptide bond connecting that amino acid chain to the amino acid chain on another NAM. So we're creating this peptide bond crosslink between these two NAMs. The enzyme transpeptidase is supposed to do that. So when this process is complete, the enzyme can leave, and it leaves behind that formed peptide bond crosslink between these two NAM monomers in the peptidoglycan cell wall structure. So this enzyme should do that job over and over and over again. It just did it here. The enzyme should then bind these chains and crosslink them. It should bind these chains and crosslink them. So how does penicillin get in the way of this process? So in our diagram, we're going to add these penicillin molecules. They're represented by this shape. And so here's a bunch of them. And this penicillin shape is just right to fit into the active site of this transpeptidase enzyme. So if we go forward one slide, here is that penicillin chemical structure sitting inside the transpeptidase enzyme active site. And if there's a chemical in an active site, that active site can no longer bind to what it is supposed to bind to. We call this competitive inhibition in biology. And so this enzyme, because the penicillin is in the way, this enzyme can no longer bind to this spot of the peptidoglycan cell wall and create that new peptide bond crosslink. So this cell wall cannot be completed when penicillin is present, because the penicillin inhibits the function of this transpeptidase enzyme. It is supposed to create those peptide bond crosslinks. And so this cell wall is left weaker. It's not as strong as it should be. And that compromised cell wall is no longer able to protect the bacterial cell wall from lysis. And these bacteria may now die. So penicillin is an amazing antibacterial drug. It was a critically important discovery when it was discovered. And we talk a lot more about that in my own microbiology course. So penicillin stops the bacterial cell from being able to make the peptide bond crosslinks in the peptidoglycan cell wall. But what about the chemical lysozyme? So lysozyme, instead of being a chemical inhibitor, lysozyme is actually an enzyme. lysozyme is an enzyme that's made by many animals, including us humans. And this enzyme performs a specific chemical reaction. So lysozyme catalyzes or performs this chemical reaction. It hydrolyzes the glycosidic bond between the NAM and the NAG residues in peptidoglycan. So hydrolysis, hydrolyzes, is just a fancy way of saying breaks, right? So lysozyme breaks the glycosidic bonds between NAMs and NAGs. So here's another representation of the peptidoglycan chemical structure. And so again, we're seeing that alternating chain of a NAG, and then a NAM, and then a NAG, and then a NAM. And again, this enzyme, lysozyme, is able to bind to this part of the chemical structure. It introduces a water molecule, so these two hydrogens and an additional oxygen are going to get input in here. And when that happens, it breaks this link between the NAM and the NAG, this glycosidic bond. So when lysozyme performs the hydrolysis, we convert the chemical structure from something like this to these independent structures where the bond holding them together is now missing, right? So lysozyme breaks the existing glycosidic bonds that exist in the peptidoglycan cell wall. Again, that makes the cell wall weaker. A compromised cell wall is not as good at protecting the bacterial cell from lysis, and those bacterial cells are more likely to die. So let's summarize, right? These two chemicals are critically important in attacking bacterial peptidoglycan cell walls, but they work in slightly different ways, right? So penicillin prevents the formation of those new peptide bond crosslinks between the NAMs within that peptidoglycan structure. We identified that penicillin is a transpeptidase enzyme inhibitor, and that when penicillin is present and able to inhibit that enzyme, the peptide bonds are missing, right? They're not able to be made, and that results in bacterial cell lysis. Meanwhile, lysozyme is an enzyme that breaks existing glycosidic bonds between NAM and NAG in that peptidoglycan structure. Again, instead of being a simple chemical that is an enzyme inhibitor, lysozyme actually is an enzyme itself. So lysozyme is an enzyme, but again, because the bacterial cell wall no longer has these important connections, this results in a weaker cell wall, and it results in bacterial cell lysis. So I hope that summary helps. As always, please let me know if you have any questions, and I look forward to talking to you again soon.